Thank you very much for this nice introduction. And I will present you our research on fast phosphorylation in FTLD. In our group, we are working on several proteins involved in neurodegeneration, and among those is also FUS, known as fusing sarcoma, or previously as TLS, translocated in liposarcoma. FUS is an RNA binding protein consisting of N-terminal low complexity domain, followed by a nuclear export signal, an RNA recognition motif, and two successive RGG domains interrupted by zinc finger, and an ending with a very typical PY nuclear localization signal. Both localization signals, so either import or export, they enable FUS to shuttle between the nucleus and cytoplasm, and under normal conditions, FUS is predominantly found in the nucleus. There it has several functions, from RNA and DNA processing, where it is involved in transcription, splicing, and repair, towards RNA protein intracellular transport, especially in large cells such as neurons, where it helps translocating RNA from the cell soma to the cell extensions, such as dendrites, so that the translation of those proteins can appear there. And it was also shown to be involved in stress response, as it was found to be one of the components of the stress granules and nuclear bodies. However, in the neurodegenerative diseases, or in those diseased neurons, first mislocalization from the nucleus into the cytoplasm can occur. And when this happens, then we face the situation of first nuclear depletion, and we are having like enhanced concentration of FUS in the cytoplasm, where FUS can form the so-called aggregates. Aggregates, as you all know, they are a common hallmark of neurodegenerative diseases, and they often cause neuron cell loss, whereas clinical presentation in those diseases often depends on the neuron type afflicted. Here, I will just mention the two of these neuron disorders, so the so-called ALS and FTLD, as in both of those FUS aggregates were noted. So we have on one side amyotrophic lateral sclerosis, ALS, in which spinal upper and lower motor neurons are majorly afflicted, leading to paralysis and death of patients within 3 to 5 years. On the other hand, we have frontotemporal dementia, in which conversely, majorly frontal and temporal cortical neurons from the brain are afflicted, and this leads to patients' cognitive decline and early onset of dementia. But as I mentioned, in both of those disorders, FUS aggregates and forms the cytoplasmic inclusions, yet those are different. So in ALS, we are facing with cytoplasmic inclusions that are mostly composed of FUS, and in this case of mutated FUS, whereas no such mutations have been detected in the majority of FTLD cases, where those FUS aggregates are composed of wild-type FUS, yet these aggregates are not homogeneous, as in ALS, but rather heterogeneous, besides FUS being composed also of other proteins of the fat family, and also consisting of a nuclear transport factor transportin 1, which was shown to bind FUS. So because we are dealing with a wild-type FUS in FTLD, the question arises, what causes this wild-type FUS to move into the cytoplasm, stays there, and form these aggregates? And it was proposed in the past that some protein misfolding, especially the one arising from post-translational modification, may be causative for that. And it was already in the 2014 that it was shown that phosphorylation of FUS, especially this low complexity region, regulates FUS aggregation. Later on, it was shown that namely recombinant FUS phase separates in vitro to form such dynamic liquid droplets, which are still soluble, but over time, those can evolve into irreversible toxic and solid aggregates. Of course, in cells, in the cytoplasm, we have also other components, such as our mRNAs, and those can partition together with Fas also in the so-called ribonucleoprotein granules, but over time, even those under prolonged stress or the mixing process can turn into those insoluble Fas inclusions. Still, in the cells, we have a chaperone protein called transportin 1, which, as said previously, binds Fas at its C-terminal region, tears it away from the aligned molecules, and brings it back into the nuclei. But as our group has shown, the phosphorylation of this C-terminal tyrosine in Fas, which is important for the transportin binding, can inhibit this transportin binding, and this leads to the impairment of the Fas nuclear localization. Namely, hydrogen bonds form between hydroxyl group of Fas and carboxylic group of aspartic acid in transportin. When tyrosine is phosphorylated, this introduces extra negative charge, leading to electrostatic repulsion of Fas and aspartic acid in transportin 1, and this way, this binding is inhibited. Online programs predicted several members of the SARC family of kinases to be involved in the phosphorylation of this last C-terminal tyrosine in Fas. And of course, for us to see how this C-terminal tyrosine phosphorylation is involved in Fas intracellular distribution, our aim was to get the phosphofas specific antibody. For this reason, pairs of phosphorylated and non-phosphorylated peptides were synthesized, and those were corresponding to the last 16 amino acids of Fas, involving either phosphorylated or non-phosphorylated tyrosine. And those were used as immunogens for this phosphofas and Fas antibody production in RABs, which was done at AMS Bio. Those antibodies were then affinity purified, ELISA and dot blot tested for cross-reactivity. And here underneath, I am showing that phosphofas only binds the phosphorylated Fas peptide, short peptide, whereas just C-terminal Fas binds both of them. But to eliminate that this phosphofas antibody is really selective only for the Fas and not for the protein of the same fat family, which all possess the PY in their sequence. We've did then lots of pull downs and Western blots, all showing that phosphofas band always appears only for when we test for the phosphofas and never when we test for any other of those fat proteins. This antibody selectivity was further confirmed by inhibitor assays, with which we then again confirmed that especially those three members of the SOC kinase family, so FIN, SOC, and ABL, indeed phosphorylate Fas by showing us this declined phosphofas signal in the cells treated with this inhibitor in concentration-dependent manner. Then to point out that this antibody detects only the last phosphorylated tyrosine in Fas and not any other within the Fas, as Fas involves or has like 36 of them. We've constructed a plasmid only of last 36 amino acids having either tyrosine or the substitute phenylalanine at the end. And as you can see, the phosphofas band only appeared in the cells co-transfected with this construct and kinases. And later on, with the phosphatase assay and with siRNA knockdown, it was shown that the phosphofas band, they always disappear either upon phosphatase treatment or siRNA knockdown. So we were wondering, okay, now how this reveals in cells in situ, how it is seen. So we've transfected the cells with kinases, and what we observed was that both FIN and SARC kinases, they give us like really very strong nuclear phosphofus appearance, whereas in case of transfecting cells with active CABL kinase, we get this, we get this strong cytoplasmic appearance of FUS. And as we know that in the cytoplasm, FUS forms those insoluble inclusions. We did then a biochemical fractionation, and what came out was that only SARC and FIN phosphorylated FUS appeared in the soluble fractions, whereas CABL phosphorylated FUS was only found in the insoluble fractions. We wondered whether this effect or whether this pattern of immunoreactivity would be similar also in cells more relevant for neurodegenerative diseases. So the experiments were repeated with a differentiated either differentiated sushi cells and NSC34 cells, and in all of those in vitro bred cell lines, the pattern of phosphofus and kinases was similar, but when using the primary neurons from mice, those revealed quite different phosphofus immunoreactivity. For example, for SARC and FIN kinases, we could not observe like really very increased phosphofus signal in either cytoplasm or nuclei compared to the non-kinase transfected cells, whereas for ABL, this signal was very much increased in the nuclei as well as in the cytoplasm of those murine primary neurons co-transfected with GFP-FUS and kinases. So with this, we wondered like what is happening then in vivo and whether or not this phosphofus is already present in mice brain. So we did a staining and we found out that the phosphofus can be detected already in the neurons of the cortex, in Purkinje cells of cerebellum, also in some separate groups of neurons in the hippocampal gyri and brainstem, whereas also some sparse astrocytes throughout the cortex also showed some weak positive signal for phosphofus. We asked ourselves whether this could be correlated also with the activity of those kinases of the SARC family. So the co-immunostaining with all three kinases and for phosphofus were performed, and we found out that the immunoreactivity for phosphofus and all three kinases, so SARC, FIN, and ABL, they nicely overlap in the hippocampus, whereas in the brainstem, only the overlap between phosphofus and SARC could be observed. Quite interestingly, in the frontal cortex, there's a very strong and granular cytoplasmic overlap was observed then also between ABL and phosphofus. And because we all know that frontal cortex is very much afflicted in FTLD, we proceeded with ABL and phosphofus towards the human samples. And those were stained first for phosphofus, and the abstaining there revealed like really diffuse, sometimes nuclear, but also cytoplasmic staining in control samples, whereas in the FTLD FUS sample, whereas in those FTLD FUS samples, we've observed such really atypical small granule inclusions. And those were really distinct from those large mature aggregates that are normally detected in those samples by commercial FUS antibodies. And then with the fluorescent immunostaining, we've which is even more sensitive, we've been able to confirm this so that the phosphofus and FUS staining only overlapped with those small granules, whereas those large aggregates, the phosphostaining did not overlap. And what was additionally observed was that in nearly 60% of the patients, the nuclear phosphofus staining was very much increased in the FTLD patients. And in those, the phosphofus staining was merely located to the nuclear envelope. Now, whether this is then correlated with the ABL kinase activity in the brains, we did the co-staining, and it was observed that active PABL kinases and phosphofus reactivity actually overlaps in those neurons of human cortex where ABL immunoreactivity really followed the pattern of the phosphofus. So phosphofus immunoreactivity was very much increased as well as ABL. To get then insight how this increased ABL activity can affect FUS or phosphofus distribution under stress conditions, we've utilized our previously generated Philippine cells expressing mSCARLY G3BP1. This protein is a component of the stress granules. We transfected those cells with the ABL kinase and exposed them to arsenides, so-called oxidative stress reagents. So what we observed was that ABL kinase induces phosphofus sequestration in stress granules. So given this overlapping pattern of CABL activity and phosphofus distribution in cortical neurons, we propose that CABL kinase is actively involved in mediating cytoplasmic mislocalization and promoting toxic aggregation of wild-type FUS in the brains of FTLD patients. So with this, I would like to end, and at the end, acknowledge and thank all the people involved in this study. So my co-workers at the Josef Stefan Institute and Faculty of Chemistry and Chemical Technology, as well as the people from Amsterdam who were providing us with the human tissues, and our neuropathologists with his students who were doing the staining on these human sections. And I thank you, and I'm happy to take questions.